February 28, 1953. The Eagle Pub in Cambridge, England. Two scientists burst through the door and one of them, Francis Crick, announces loudly that they've discovered the secret of life. The other patrons probably think he's had too much to drink, but Crick and his collaborator James Watson have just solved the structure of DNA, the molecule that carries genetic information in every living thing. This discovery will transform biology, revolutionize medicine, create entire industries, answer humanity's deepest questions about heredity and evolution, and raise profound ethical dilemmas we're still grappling with today. This is the story of how scientists unraveled life's most fundamental mystery, a story of brilliant insights and bitter rivalries, overlooked contributions and stolen credit, patient accumulation of evidence and sudden breakthroughs. It's the story of how humanity learned to read the instruction manual written inside every cell. The story begins long before anyone knew DNA existed. For millennia, people observed that children resemble parents, that traits run in families, that you can breed animals and plants for desired characteristics. But how heredity worked remained mysterious. Various theories proposed that male semen contained miniature humans that simply grew larger in the womb, or that characteristics from both parents blended like mixing paint. None of these theories were correct. The foundation of modern genetics was laid by an obscure Austrian monk named Gregor Mendel. In the 1850s and 1860s, Mendel conducted meticulous experiments breeding pea plants in his monastery garden. He crossed plants with different traits, tall and short, green seeds and yellow seeds, smooth and wrinkled, and carefully counted offspring. He discovered that traits didn't blend but were inherited as discrete units according to mathematical ratios. Mendel proposed that each parent contributed a factor for each trait, and that some factors were dominant over others. A plant with one tall factor and one short factor would be tall, but could still pass the short factor to offspring. Mendel had discovered what we now call genes, though he didn't know their physical nature. Mendel published his findings in 1866, but they were ignored. The scientific community wasn't ready for mathematical analysis of biology. Mendel died in 1884, his work unrecognized. Only in 1900 were his papers rediscovered independently by three botanists, sparking the science of genetics. But what were genes made of? Where were they located? How did they work? These questions drove biology for decades. Chromosomes, thread-like structures in cell nuclei, were discovered in the 1880s. Scientists observed that during cell division, chromosomes duplicated and were distributed equally to daughter cells. In the early 1900s, several researchers proposed that chromosomes carried hereditary information. Thomas Hunt Morgan's fruit fly experiments at Columbia University in the 1910s proved this conclusively, showing that specific traits were linked to specific chromosomes. But chromosomes contain two major components, proteins and nucleic acids. Most scientists assumed proteins carried genetic information. Proteins were complex molecules with 20 different amino acids that could be arranged in endless combinations. Nucleic acids, by contrast, seemed simple and repetitive. How could something so simple encode life's complexity? The nucleic acid in question, deoxyribonucleic acid, or DNA, had been discovered in 1869 by Swiss physician Friedrich Miesser. Studying white blood cells from pus on surgical bandages, Miesser isolated a novel substance from cell nuclei. He called it nuclein. Chemical analysis showed it contained phosphorus and nitrogen but no sulfur, distinguishing it from proteins. Miesser recognized nuclein was important but didn't know its function. For decades, DNA was considered a structural molecule, perhaps a scaffolding for the important proteins. This tetranucleotide hypothesis proposed that DNA was a monotonous repeating structure of four bases, adenine, thymine, guanine, and cytosine, in equal proportions. Such a simple, repetitive molecule couldn't possibly carry complex genetic information. The breakthrough came from an unexpected source, bacteria. In 1928, British bacteriologist Frederick Griffith conducted experiments with two strains of Streptococcus pneumoniae, one virulent, one harmless. He killed the virulent bacteria with heat and mixed them with live harmless bacteria. Somehow, the harmless bacteria were transformed into virulent ones, and this trait was inherited by their descendants. Something from the dead virulent bacteria had transformed the living harmless bacteria. But what was the transforming principle? In 1944, a team at Rockefeller University led by Oswald Avery, with Colin MacLeod and Maclyn McCarty, identified it. Through painstaking biochemical experiments, they systematically eliminated possibilities. They treated the transforming extract with protein-destroying enzymes. It still worked. They destroyed lipids and carbohydrates. Still worked. Only when they added enzymes that destroyed DNA did the transformation fail. The transforming principle was DNA. This should have been revolutionary. Avery had proven that DNA, not protein, carried genetic information, but the scientific community was slow to accept it. Avery was cautious in his claims, and many remained skeptical. Proteins were still favored as the genetic material. Definitive proof came in 1952 from Alfred Hershey and Martha Chase's elegant experiment with bacteriophages, viruses that infect bacteria. They labeled phage proteins with radioactive sulfur and phage DNA with radioactive phosphorus. When phages infected bacteria, only the radioactive phosphorus entered the bacterial cells. The protein coat stayed outside. Yet new phages were produced, proving DNA alone carried the genetic instructions. The Hershey-Chase experiment convinced skeptics DNA was the genetic material. But knowing DNA carried genetic information didn't explain how it worked. What was DNA's structure? How did it encode information? How did it replicate so each cell received an exact copy? By the early 1950s, several facts were known. DNA was a long polymer, a chain of repeating units called nucleotides. Each nucleotide contained a sugar molecule, deoxyribose, a phosphate group, and one of four nitrogenous bases, adenine A, thymine T, guanine G, or cytosine C. The sugar and phosphate formed the molecule's backbone. The bases were the variable part. Crucially, in 1950, Austrian-American biochemist Erwin Chargaff published findings that destroyed the tetranucleotide hypothesis. Analyzing DNA from different organisms, Chargaff found that while the amounts of the four bases varied between species, within any species the amount of adenine always equaled thymine, and guanine always equaled cytosine. These became known as Chargaff's rules. The pattern suggested DNA's structure had a specific organization, though Chargaff himself didn't figure out what it was. X-ray crystallography, using X-rays to determine molecular structures, offered a way forward. English physicist Maurice Wilkins at King's College London was using this technique to study DNA. He was joined in 1951 by Rosalind Franklin, a brilliant experimentalist with expertise in X-ray crystallography. Franklin was meticulous and demanding, producing the clearest X-ray diffraction images of DNA ever obtained. Her Photo 51, taken in May 1952, showed a distinctive X pattern indicating a helical structure. Franklin's images contained crucial information about DNA's structure, but interpreting them required complex mathematical analysis. Franklin's relationship with Wilkins was contentious. She thought she'd been hired to lead the DNA project. Wilkins thought she was his assistant. Both were stubborn and poor communicators. They barely spoke, creating a toxic work environment. This dysfunction would have tragic consequences. Meanwhile, at Cambridge University's Cavendish Laboratory, two scientists were obsessed with DNA structure. James Watson was a 23-year-old American biologist, brilliant but arrogant and socially awkward. Francis Crick was a 35-year-old English physicist who had come to biology late, still working on his PhD. They were an odd pair, Watson young and impatient, Crick older and voluble, but they complemented each other perfectly. Watson and Crick weren't experimentalists. They built models, physical models using metal plates and wire, trying different arrangements until one fit the known facts. Their approach was borrowed from Linus Pauling, the American chemist who had discovered the alpha helix structure of proteins using model building. In 1951, Watson attended a lecture by Franklin describing her X-ray results. Watson, not a trained crystallographer, misunderstood some technical details and returned to Cambridge with garbled information. He and Crick built a model with the phosphate sugar backbone in the center and bases on the outside. They invited Franklin and Wilkins to see it. Franklin immediately recognized it was wrong. Her data showed the backbone must be on the outside. Watson and Crick were humiliated. Their supervisor, Lawrence Bragg, banned them from working on DNA, telling them to focus on proteins instead. They agreed but kept thinking about DNA. The race intensified when they learned Linus Pauling was also working on DNA structure. Pauling, perhaps the world's greatest chemist, was formidable competition. In January 1953, they learned Pauling had proposed a triple helix model with the backbone in the center, similar to their failed model. Pauling was wrong, but he'd realize it soon and correct his mistake. Then came the crucial break. In late January 1953, Morris Wilkins showed Watson Franklin's Photo 51 without Franklin's permission or knowledge. Seeing the clear X pattern, Watson immediately recognized it indicated a double helix. He rushed back to Cambridge. Watson and Crick also received, again without Franklin's knowledge, her detailed research report analyzing DNA structure. Max Perutz, a crystallographer at Cambridge, had received the report as part of a government committee and shared it with Crick. This report contained precise measurements and analysis that Franklin had painstakingly derived. Armed with Franklin's data, obtained unethically, without her consent, Watson and Crick returned to model building with renewed urgency. They knew DNA was a double helix with the backbone outside and bases inside, but how were the bases arranged? The key insight came from Chargaff's rules. A always equaled T, G always equaled C. What if the bases paired? What if A always bonded with T and G with C? Jerry Donohue, an American crystallographer sharing their office, pointed out that the chemical forms of the bases Watson was using were wrong. With the correct forms, Watson realized A and T fit together perfectly, as did G and C. The base pairs were the same width, creating a uniform diameter for the helix. It was elegant. Two strands running in opposite directions wound around each other like a twisted ladder. The sugar phosphate backbones formed the sides. The base pairs A with T, G with C formed the rungs. The bases were inside, protected. The specific pairing meant each strand was complementary to the other. If you knew one strand's sequence, you knew the others. More importantly, the structure immediately suggested how DNA replicated. The two strands could unwind, and each could serve as a template for building a new complementary strand. This explained how genetic information was copied precisely during cell division, one of life's fundamental mysteries. Watson and Crick published their findings in Nature on April 25, 1953. The paper was just one page, a model of scientific brevity. They described the double helix structure and noted, with famous understatement, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." The same issue contained papers by Wilkins and Franklin, providing the X-ray evidence supporting the model. Franklin's paper was published third, and her contribution was downplayed. Watson and Crick thanked her only briefly and didn't mention they'd seen Photo 51 or her detailed report without permission. The double helix was immediately recognized as a monumental discovery. It explained how genetic information was stored, replicated, and potentially translated into proteins. It provided a molecular foundation for understanding heredity, evolution, and life itself. But Rosalind Franklin never received proper credit during her lifetime. She died of ovarian cancer in 1958 at age 37, likely caused by radiation exposure from her X-ray work. She never knew Watson and Crick had used her data without permission. The Nobel Prize in Physiology or Medicine for the discovery of DNA structure was awarded in 1962 to Watson, Crick, and Wilkins. The Nobel Prize isn't awarded posthumously, so Franklin was excluded. But even if she'd been alive, she likely wouldn't have been included. Her contribution was systematically minimized. Watson's 1968 memoir, The Double Helix, portrayed Franklin unflatteringly as Rosie, a name she never used, describing her as difficult, unattractive, and incompetent. This portrayal shaped public perception for decades. Only later did historians recognize Franklin's crucial contributions and the unethical way her data was obtained. Today she's recognized as a pioneering crystallographer whose work was essential to discovering DNA structure. The discovery opened new frontiers. Scientists immediately began working out how DNA encoded proteins. The genetic code, how sequences of bases specified amino acids in proteins, was cracked by 1966 through work by Marshall Nirenberg, Har Gobind Khorana, and others. They found that 3 base sequences, codons, specified each amino acid. With 4 bases, there were 64 possible 3-base combinations, more than enough to encode 20 amino acids. The structure of DNA also explained mutations. Errors in copying, wrong bases inserted, would change the genetic code and potentially alter proteins. Radiation or chemicals could damage DNA, causing mutations. This explained how evolution worked at the molecular level and why radiation caused cancer. Recombinant DNA technology, developed in the 1970s, allowed scientists to cut and paste DNA sequences, inserting genes from one organism into another. In 1973, Herbert Boyer and Stanley Cohen created the first genetically modified organism by inserting foreign DNA into bacteria. This launched genetic engineering and biotechnology industries. Genetic engineering raised ethical concerns. Should we modify organisms, create new life forms? The potential benefits were enormous, producing medicines, improving crops, treating diseases. But so were the risks, ecological damage, unforeseen consequences, playing God. Scientists meeting at Asilomar in 1975 established guidelines for recombinant DNA research, a rare example of scientists proactively addressing their technology's risks. DNA fingerprinting, developed by Alec Jeffreys in 1984, revolutionized forensics and paternity testing. Each person's DNA is unique, except identical twins. So DNA samples from crime scenes could identify perpetrators with extraordinary accuracy. DNA evidence has both convicted the guilty and exonerated the innocent, including people on death row. The Human Genome Project, launched in 1990, aimed to sequence all 3 billion base pairs in human DNA. It was biology's moonshot, an international effort costing billions. The project competed with private company Celera Genomics, led by Craig Venter. In 2000, both announced they'd completed draft sequences. The full sequence was published in 2003, 50 years after the double helix discovery. The human genome revealed surprises. Humans have only about 20,025,000 genes, far fewer than expected. Much of the genome doesn't code for proteins, junk DNA, though some has regulatory functions. Humans share 99.9% of their DNA. Genetic differences between individuals are tiny. We share 98–99% with chimpanzees, confirming Darwin's theory of common ancestry. Genomics transformed medicine. Knowing disease genes allows testing for genetic conditions. Personalized medicine tailors treatment to individual genetic profiles. CRISPR-Cas9, discovered in 2012, allows precise gene editing. It could cure genetic diseases by correcting faulty genes, but also enables designer bobbies, selecting traits like intelligence or appearance. Chinese scientist He Jianqiu's 2018 creation of gene-edited bobbies sparked global outrage and raised questions about boundaries. DNA analysis revolutionized our understanding of human origins and migration. Analysis of mitochondrial DNA, inherited maternally, showed all humans descend from a woman in Africa roughly 150,000 to 0,000 years ago, mitochondrial Eve. Y chromosome analysis traced paternal lineages. DNA from archaeological remains revealed that modern humans interbred with Neanderthals and Denisovans. We're not pure Homo sapiens, but hybrids carrying ancient human DNA. Ancient DNA techniques resurrected extinct species genomes. Woolly mammoth genomes have been sequenced from frozen remains. De-extinction, bringing back extinct species like mammoths or passenger pigeons, moved from science fiction to theoretical possibility, though ethical and practical questions abound. DNA databases raised privacy concerns. Governments collect DNA from criminals, but should DNA be collected from everyone? Insurance companies want genetic information to assess risk. Employers might discriminate based on genetic predispositions. The Genetic Information Non-Discrimination Act, 2008, banned genetic discrimination in America, but enforcement is imperfect. Ancestry testing companies like 23andMe and AncestryDNA made genomics consumer-friendly. For $99, you could learn your ancestry and genetic health risks. Millions submitted DNA, creating massive databases. Police used these databases to solve crimes. The Golden State Killer was caught by matching crime scene DNA to relatives in genealogy databases. This was brilliant detective work, but raised questions about consent. People uploaded DNA for ancestry, not criminal investigations. Synthetic biology takes genetic engineering further, designing organisms from scratch. Researchers created the first synthetic bacterial genome in 2010. Could we design organisms to produce biofuels, clean pollution, or manufacture materials? Could synthetic biology create pandemic pathogens? The dual-use potential, beneficial and dangerous applications, requires careful oversight. DNA computing uses DNA's information storage capacity for computation. DNA can store vast amounts of data in tiny volumes. Researchers encoded an entire book in DNA. Biological computers using DNA could theoretically outperform silicon computers for certain tasks. The discovery of DNA's structure also profoundly impacted philosophy and culture. We learned that life's complexity emerges from simple chemical rules, that heredity, evolution, and development all follow from DNA's properties. This molecular reductionism was intellectually satisfying but also troubling. Are we just machines executing genetic programs? What about free will, consciousness, and humanity's specialness? DNA entered popular culture. Movies like Jurassic Park, 1993, brought cloning and genetic engineering to mainstream awareness with dubious accuracy. CSI-type shows made DNA evidence familiar. DNA metaphors permeate language. We speak of company DNA or DNA of cities. The double helix became an instantly recognizable scientific icon. Watson's later career was controversial. He made increasingly offensive statements, particularly about race and intelligence, promoting discredited notions that genetics explain group differences in IQ. In 2007, he claimed Africans were less intelligent than Westerners due to genetics. These statements, lacking scientific support and contradicting genetics' actual findings, damaged his reputation. Cold Spring Harbor Laboratory, which he'd led for decades, stripped his honorary titles in 2019. Crick, who died in 2004, remained a respected scientist. He contributed to understanding the genetic code and consciousness. Wilkins, who died in 2004, had a quieter career. Franklin's legacy has been rehabilitated. She's now recognized as a pioneering woman in science whose contributions were stolen and minimized by sexism. The double helix discovery was science at both its best and worst. At its best, brilliant problem-solving, elegant theory fitting data beautifully, opening vast new fields. At its worst, credit stolen from a woman scientist, unethical use of data. Arrogance and bad behavior rewarded, while meticulous experimental work was undervalued. DNA's discovery demonstrated that life, despite its complexity, follows understandable chemical principles. It unified biology, explaining heredity, evolution, development, and disease at the molecular level. It created biotechnology and genomics industries worth hundreds of billions. It transformed medicine, agriculture, and forensics. But it also raised profound questions still being grappled with. Should we edit human embryos, create designer bodies, resurrect extinct species, modify organisms to solve problems like climate change? Who owns genetic information, individuals or whoever sequences it first? These aren't just scientific questions but ethical, legal, and philosophical ones. From Mendel's pea plants to CRISPR gene editing, from Friedrich Miescher's Nuclein to the Human Genome Project, from that momentous February day in the Eagle Pub to today's genome sequencing in hours for hundreds of dollars, DNA's story is one of humanity's greatest intellectual achievements. The double helix explained life's most fundamental mystery, how biological information is stored, copied, and passed to offspring. It revealed that all life on Earth shares a common chemical language. Written in four-letter alphabet of A, T, G, and C, it showed that humans and bacteria, oak trees and mushrooms, elephants and insects all use the same genetic code, powerful evidence for common ancestry and evolution. Yet despite 7 decades of study, DNA still holds mysteries. How does the same genome produce different cell types? How do genes and environment interact? What does most of the genome actually do? How does DNA encode not just proteins but complex traits like intelligence, personality, and behavior? Can we understand consciousness by understanding the brain's genetic programming? The discovery of DNA's structure was a beginning, not an end. It opened a door that scientists are still walking through, discovering new rooms, CRISPR, epigenetics, synthetic biology, ancient DNA, that Watson and Crick never imagined. When Francis Crick announced, "We've found the secret of life in that Cambridge pub," he was both right and wrong. Right that DNA's structure was life's fundamental code. Wrong that discovering it would answer all questions. Instead, it revealed how much we didn't know and opened up entirely new categories of questions. The double helix is perhaps science's most famous image, elegant, simple, beautiful. It reminds us that nature's deepest secrets often have a simplicity and symmetry that feels almost inevitable once discovered. The structure of DNA was there all along, waiting in every cell of every living thing, patiently waiting for humans to figure it out. From Gregor Mendel's garden to the Human Genome Project, from Rosalind Franklin's X-ray images to CRISPR's gene scissors, the discovery and understanding of DNA represents one of humanity's supreme intellectual achievements. It's changed how we understand ourselves, where we came from, what we might become, and what it means to be alive.